So, part 2 of protein synthesis is where we take the mRNA that was produced in the first stage, transcription, and use the sequence of bases that's in the mRNA to determine the sequence of amino acids in a polypeptide. So, just to recap what we did in the last tutorial, transcription is where we saw the importance of the RNA nucleotides, complementary base pairing with the nucleotides on the DNA coding strand, the importance of the hydrogen bonds holding those complementary bases together, the importance of the enzyme RNA polymerase joining those nucleotides together, and the importance of the process of splicing to remove those non-coding regions, the introns, so that you have a mature mRNA that's made up entirely of coding regions. So this little tutorial is all about how we get the sequence of bases in the mRNA to be a sequence of amino acids. And there's another type of RNA that you need to be aware of. So this little picture shows this type of RNA is called transfer RNA, and we're going to call it tRNA from now on. And it's basically one strand of RNA folded over like this. I haven't drawn in all the bases this time, apart from these three at the bottom. These are important. And there's a point of attachment at the top for an amino acid, a specific amino acid determined by the bases at the bottom to attach. Yeah, so this amino acid attaches to this transfer RNA, and a different amino acid will attach to a different tRNA. So these bases, these three bases at the bottom, they're called an anticodon. And they're important because they allow the transfer RNA to interact with the mRNA, which I've drawn down here. Now, if that's the anticodon, we can say that this triplet of bases here is called a codon. And in fact, a codon is just a triplet of bases that codes for one amino acid. That's why it's called a codon, because it's coding for an amino acid. And this particular codon, AUG, codes for the amino acid methionine, for instance. So this anticodon can come in and complementary base pair with this codon. They'll come together and they'll form hydrogen bonds between the complementary bases. So we can write in which bases these would be. If that's A, the complementary base on the anticodon would be U. If that's U, then that complementary base will be A. And that's G, so that's going to be C. So the anticodon will always be complementary to the codon. So with that little bit of background, we can go back to our picture of translation. And here we have a strip of mRNA, and we have the mRNA attached to a ribosome. Now my ribosome is actually made up of two subunits, a small subunit and a big subunit, and the mRNA fits in between them, if you like. The ribosome is there to hold the mRNA in place, to hold all the machinery that's involved in putting the amino acids in the right order in place. If we just go back to this diagram here, we know our ribosomes are little dots because they're so tiny, but actually if you were able to magnify them enormously, you'd be able to see that they were made up of these two subunits, and that's so that the mRNA can fit in between. So every three bases on the mRNA codes for one amino acid. You probably remember that from GCSE. 3 bases on the mRNA codes for one amino acid. And of course, we've just heard that that's called a codon. Okay, and we just refer back to our little picture here, we just saw that the amino acid comes and knows where to position itself because of the tRNA. So let's draw that into this little picture here. The tRNA comes along. The tRNA with the complementary anticodon comes along, and complementary base pairs with this codon. Okay, so it literally, I've drawn it kind of up in the air, but it will literally come in and steady itself there. And then the next tRNA will come in and it will have a anticodon that is complementary to the codon here. This is going to be G, that one will be U, that will be A, and it will hold the amino acid in place. And then the next tRNA comes along and carrying an amino acid, and the next codon will be, anticodon will be complementary to this codon, so that'll be C, C and A. And eventually what will happen is these amino acids will be joined together by peptide bonds. And eventually what will happen is the amino acids will be dropped off, the tRNA molecule will go off to find another amino acid, and the amino acids left behind will be all joined together, and eventually you'll have a long chain of amino acids joined together by peptide bonds. We have a polypeptide, a primary structure protein. Now what's important is that this sequence of amino acids has been determined by the sequence of bases on the mRNA. Just say that again, that's quite important. So the sequence of amino acids, the order in which the amino acids are joined together in the polypeptide, has been determined by the sequence of bases in the mRNA. And of course, we learned earlier that the sequence of bases in the mRNA is determined by the sequence of bases in the DNA. So what's really happening is that this sequence of bases in the DNA is determining the sequence of amino acids in the polypeptide. So one more thing, I haven't drawn in all the bases here, but there's one codon at the end of the mRNA that doesn't code for an amino acid. So it signals the stop of that translation, and we call this a stop codon. So everything that's involved in translating, the enzymes that are involved, the ribosome that's involved, all the tRNA molecules, everything is released at this point and the translation stops. Okay.